会议呢，基本上是一个科学的会议，是一个有关科学发生的历史。
Signal transduction and active evaders of transcription.
Yeah.
ST8.
I hope I can see the slides. I'm going to have to ask you to change them for me, please.
We published the first analysis.
So I'll do my best to give my talk remotely in California.
And mechanisms of signaling and physiological fine-tuning.
Virus and receptor discovery and development of vaccine antibody.
而这个交往更多的是在胁迫感染这种情况下发生的一个死亡。
Okay, can you see it now?
Yes. That's perfect now.
Hang on. Okay, let's see. Okay, so here we go. I'm sorry that I can't be there in person to share in this great celebration of the discovery of the JAK-STAT signaling pathway. So I'll do my best to give my talk remotely in California. So I thought I would start with a brief history of protein phosphorylation that ultimately led up to the discovery of tyrosine phosphorylation and then the JAK-STAT signaling pathway. So covalent linkage of phosphate to protein was discovered in 1906 by Levine and Alsberg. In fact, they reported that vitelline, an egg protein, contains covalent phosphate in the second volume of the JBC in 1906. The same group went on to demonstrate that the phosphate was linked to serine in vitelline about 25 years later. The next major event was the discovery of enzymes that could catalyze transfer of phosphate from ATP onto proteins. The work of Eugene Kennedy in 1954, and then the seminal work of Ed Krebs and Eddie Fisher in 1959 with the discovery of phosphorylase kinase, and then Ed Krebs' discovery of the cyclic AMP-dependent protein kinase in 1968. Surprisingly, then the next phosphoamino acid to be discovered was phosphohistidine, the work of Paul Boyer, who showed that succinyl-CoA synthase, a mitochondrial enzyme, uses a phosphohistidine intermediate, but very little was done on histidine phosphorylation subsequently, although we have been, and many others have been working on this more recently as a protein modification. And then we stumbled on tyrosine phosphorylation in 1980, and I'll tell you briefly about that discovery in a few minutes. And ultimately, all of this then led to the approval of the first tyrosine kinase inhibitor, Gleevec, in 2001. So if we look at the discoveries and the history of tyrosine phosphorylation, the first papers began to be published in 1979 and 1980 from our work, and also the work of Owen Whitty and David Baltimore and Stanley Cohen on the ABL and EGF receptor tyrosine kinases. I would say the next important event was the identification of the sarcomology 2 domain by Tony Paulson's group, and subsequent work showing that the SH2 domain binds phosphotyrosine. I thought I would just remind you all that the sarcomology 2 domain, the SH2 domain, is nearly 40. Tony Pawson's seminal paper in Mole Cell Biol in 1986 reported this new domain that was conserved in several tyrosine kinases in addition or upstream of the catalytic domain. You can see in this paper here, which I looked up, has been cited nearly 900 times since then. And this culminated in Tony Pawson's and Stefan Knapp's work on the structure of the Phipps tyrosine kinase with the SH2 domain interacting with the catalytic domain. So in terms of the meeting's theme, of course, there were key events in the discovery of the JAK-STAT pathway, particularly Andrew Wilkes's discovery of the JAK1 and JAK2 tyrosine kinases in his seminal PNAS paper in 1989. And then obviously the terrific foundational work from Fu and Darnell's groups showing that the JAK kinases phosphorylate the STAT latent transcription factors, resulting in nuclear translocation and induction of gene expression. Of course, that was the work of many people. And then, importantly, the first JAK inhibitor for therapeutic purposes was approved now nearly 13 or 14 years ago, ruxolitinib. So we stumbled on tyrosine phosphorylation in 1979. At the time, I was interested in how this small DNA tumor virus, polyomavirus, causes transformation of fibroblastic cells in culture and tumors in rodents like hamsters. And it's a very simple virus, the 6 genes, it's a circular 5 kilobase pair genome. And the early region, which is transcribed immediately the virus nucleus, encodes 3 overlapping proteins, the T antigens. And a middle-sized T antigen, 60,000 Daltons, was shown to be capable by itself of transforming fibroblasts in culture. And here I am in 1979 with a dark brown beard instead of my current white beard. And we were wondering how this single protein was able to convert cells from a normal state into a tumor state. And we followed up on an observation made by Mark Collette and Ray Erickson, who'd shown that the transforming protein of the Rous sarcoma virus, an RNA tumor virus called BSAR, had an associated protein kinase activity. And that was really striking because one could imagine that a protein kinase, which could phosphorylate many protein substrates, might affect many processes in the cell, including morphology and proliferation. And so many of us working on different tumor viruses decided to test whether the transforming proteins of these viruses might also have protein kinase activity. So we set out to test whether polyoma middle T antigen has an associated kinase activity. So we did this by immunoprecipitating the T antigens with an anti-tumor serum and then incubating the immunoprecipitate with gamma p32 ATP to do an in vitro kinase assay. And you can see here in this autoradiogram on the left that the middle T band itself, the 60K band itself, became labeled with p32, even though it was barely labeled in vivo with p32. Now, as controls, we had mutants of polyomavirus which lacked transforming activity, but which still expressed in some cases, like the NG59, the T antigen, and we noted that these transforming mutants lacked this kinase activity. So two other groups, Alan Smith's group and Tom Benjamin's group at ICRF and Harvard, respectively, had also found that polyoma middle T has an associated kinase activity, and we all presented our findings at the 1979 Cold Spring Harbor Symposium on tumor viruses in May of 1979. We discussed our common results and decided to co-submit our papers to Cell when we got back. And our paper was submitted the old-fashioned way, in other words, by mail, on July 11th, 1979. So I realized that the reviewers would probably ask what amino acid was getting phosphorylated in middle T in this in vitro kinase reaction. And so a couple of days later, you see on June the 14th, I set out to determine the amino acid specificity by cutting out the P32 labeled middle T band from a gel and hydrolyzing it in 6-normal HCl to identify phosphoamino acids that were released from the protein. And so we expected it to be either phosphoserine or phosphothreonine, which were the only known phosphoamino acids. And so I mixed into this acid hydrolysate marker, phosphoserine and phosphothreonine, which we could detect with ninhydrin staining. And then the plate was, the samples were separated by a thin layer electrophoresis on a cellulose plate, pH 1.9. And following the electrophoresis, the plate was dried and stained with ninhydrin, and you can see these circles here, which are phosphoserine and phosphothreonine. The plate was exposed to an X-ray film, and an overnight exposure showed this, not very much signal there. With the eye of faith, I thought I could see a faint smudge here, which wasn't either phosphoserine or phosphothreonine. With a longer exposure, it was clear there was a spot there. It wasn't phosphoserine or phosphothreonine. But luckily, as an undergraduate in Cambridge, in my biochemistry course, I was taught the structures of all 20 amino acids. And here in 1962, I drew out the structure of tyrosine. And so I knew there was a third hydroxy amino acid in addition to serine and threonine. And I guessed that this spot might be phosphotyrosine. And I made some marker phosphotyrosine and ultimately could prove that this spot was phosphotyrosine, which was totally unexpected, never been reported as a protein modification. So that was a stroke of luck, if you want. But at the same time in the lab, we were working on the Ras sarcoma virus, VSARC kinase, and one day I ran out a SARC kinase reaction, which actually phosphorylates the heavy chain of the antibody that precipitates SARC. Again, P32 labeled, and much to my amazement, the SARC kinase reaction also generated phosphotyrosine. So I should actually tell you that Ericsson's group had reported that SARC was a threonine kinase, and I'll tell you why in just a second. They got that wrong. So we resubmitted our paper to Cell in response to the reviewer comments, which indeed said, what is the amino acid getting phosphorylated? And it was published in the December issue of Cell in 1979, where we had included these new data showing that the middle T kinase phosphorylated tyrosine. And we reported the evidence that the SARC protein is a tyrosine kinase in PNAS, and in March of 1980. And in this case, we could show that cells infected by rosarcoma virus have high levels of phosphotyrosine in protein that is rapidly diminished if you have a temperature-sensitive mutant of rosarcoma virus when the cells are shifted to 42 degrees. Now, amazingly, all the experiments in the Cell and PNAS papers were done in less than 5 months, and all of the experiments in the PNAS paper were done in less than a month. If anyone's interested, I wrote a little blow-by-blow account of the experiments in the PNAS paper. So it turned out that the discovery of phosphotyrosine was really due to my laziness in not making up fresh pH 1.9 electrophoresis buffer. And on repeated use, its pH had dropped from pH 1.9 to pH 1.7. And this fortuitously resulted in the separation of phosphotyrosine from phosphothreonine. And Ray Erickson's group had used fresh pH 1.9 buffer, which is why they had missed this. I should also point out that while the tyrosine kinase activity of the VSARC protein is intrinsic, in other words, it is a kinase, polymer middle T lacks kinase activity by itself, but is associated with the cellular SARC protein, which is how it transforms cells. So over the years, we've come to realize that post-translational modifications are a mechanism to increase proteome complexity. We start with around 20,000 coding genes. We can probably generate, by splicing, about 100,000 proteoforms. But if one now uses different post-translational modifications combinatorially, one can generate perhaps 1 million protein states. Now, there are a lot of different post-translational modifications. Nearly 400 have been reported. But phosphorylation is undoubtedly the most prevalent. And I just show you these numbers from the Cell Signaling Phosphocyte Plus database, which lists nearly 300,000 unique serine, threonine, and tyrosine phosphocytes in the human proteome. And of these, 45,000 are phosphotyrosine sites. So, to sort of summarize then, the finding that the VSARC and the cellular SARC protein, which we also showed, phosphorylate tyrosine, gave us the first tyrosine kinases in 1979. By the end of 1980, 4 different tyrosine kinases were known. VSARC could be able from the work of Whitty and Baltimore, the EGF receptor from Stanley Cohen's group, and the VFIPS-FES from the work of Tony Pawson and Chuck Shear. Now, about 40 years ago, the first protein kinase sequences began to be reported. These 3 serine threonine kinases, the cyclic P-dependent protein kinase, cyclic G-dependent protein kinase, and phosphorylase kinase were all sequenced the old-fashioned way. In other words, the protein was sequenced. And then this group on the top of kinases were all the oncogenic tyrosine kinases, including SARC, FES, and ABL, which was sequenced from the cDNAs generated from the viral RNAs. And I began to align these by I. BB stands for before blast, and it became apparent that there were motifs, short motifs, along the length of the catalytic domain that were conserved in all the kinases, both the serine, threonine, and the tyrosine kinases. One particularly interesting motif is this HRDL motif, which is distinct between the tyrosine kinases here and the serine, threonine kinases. And so this RDL motif can be used to predict whether a protein kinase is a tyrosine kinase or a serine, threonine kinase. You might be amused to know that the first protein kinase sequence alignment wasn't published in a regular journal, but instead in the Scientific American Here's the figure in an article I wrote on the proteins of oncogenes in 1984. So there are 12 of these short motifs along the 300 residue catalytic length of the 300 residue catalytic domain. The catalytic domain can either be an almost entire protein, like the CDC2 or pKa, or part of a much larger protein, as is the case with the EGF receptor. And it turns out that these motifs, like the DFG motif and the RDL motif, which has the catalytic base, the aspartate there, line the catalytic cleft. And here you can see in this structure from Susan Taylor's group of pKa with a bound ATP molecule with the phosphate ready to be transferred. That they all are lining this cleft where ATP and substrates bind. And when they be mutated, all of these motifs are essential for catalytic activity. So with the ability to search newly reported cDNA sequences, ESTs, it became apparent there would be a large number of protein kinase genes, and also the use of PCR with oligonucleotides targeting some of the conserved motifs. The number of kinases began to expand very rapidly, and I should point out that Andrew Wilkes' paper in PNAS in 1989 was the first paper where PCR was used to clone novel protein kinases. So in 1987, when the number of kinases began to rise almost exponentially, I predicted there might be as many as 1,001 protein kinases. It turns out that that was a slight overestimate. The human kinome, the collection of all protein kinases, has only around 530 protein kinases, but there are kinomes like the eucalyptus kinome that grows outside our house here, has 2,500 protein kinases. When the human genome sequence was reported in 2001 by the Ventnor Celera group and by the Francis Collins NIH group, we could predict the entire human proteome and deduce how many of these might be protein kinases. And largely through the efforts of Gerard Manning at Sujian, he was able to identify around 478 EPKs, eukaryotic protein kinases, that could be displayed on this unrooted tree with 7 major branches, the tyrosine kinases on the top. The basophilic kinases, including protein kinase C, protein kinase A, and protein kinase G, calcium calmodulin regulated kinases, and the proline-directed kinases like the MAP kinases and the CDKs. Oops. There were a handful of atypical protein kinases known at the time or reported at the time. Not all of these have stood the test of time. Particularly important are the PI kinase-related kinases, which have an atypical kinase domain but related to the EPKs. These include ATM, ATR, DNA PK, and SMUG1, all of which recognize nucleic acids, and mTOR, the energy sensing kinase. Interestingly, about 10% of these protein kinases missed one or more of the catalytic residues and have been classified as pseudokinases, and many of these have scaffolding functions. We fast forward now another 16 years, the number of kinases increased slightly, no more traditional EPKs, but some about 20 new remote or atypical kinases, including PAM20C, which is the real casein kinase, and the enemy family of histidine kinases that I won't be discussing. So if you want another view of the tree, here is me with my beard dribbling kinases out of the bottom. drawn by a speaker at a meeting I attended in 1990. So as I said, by the end of 1980 there were 4 tyrosine kinases known. By the time the human genome sequence was reported, we could predict there are 90 human tyrosine kinases. So about a quarter of all protein kinases. And here they are. This is the tyrosine kinase branch of the tree, of 90 of them. Actually, only 78 of them are active, and 58 out of the 90 tyrosine kinases are receptor tyrosine kinases. Here was the first tyrosine kinase SAR. It's in a family of 8 related cytoplasmic tyrosine kinases, and here, of course, are the all-important JAK family kinases, JAK1, 2, 3, and TYK2. So over half of these tyrosine kinases are implicated in cancer through activating mutations or overexpression. And overall, around a third or more than a third of protein kinases in the human kinome are implicated in disease. Many of these in cancer, but also there are hereditary mutations in immune regulatory genes that cause immunodeficiency and in kinases that are important for development. The finding that several of the tyrosine kinases first identified drove cancer led to interest in developing inhibitors of these protein kinases, and currently there are 124 small molecule kinase inhibitors that have been clinically approved either in the US or in other countries in the world, of which 86 are tyrosine kinases. And here's a timeline of the approval of both protein kinase, antibody kinase inhibitors like herceptin, and small molecule inhibitors like Gleevec, which was the first small molecule inhibitor to be approved in 2001. And you can see that over the last 23 years there's been an almost exponential increase in the number of small molecule kinase inhibitors approved. And here are some that have been reported in the last year or two, not in this graphic here, including the first allosteric kinase inhibitor, asciminib, and deucravacitinib, also known as TYK2. Now, of course, among these 80 or so kinase inhibitors are several JAK kinase inhibitors, starting with ruxolitinib in 2011, and then tofacitinib in 2012, and then an avalanche of JAK inhibitors, starting with upadacitinib in 2019. You can see there have just been several approved in the last 3 years, and it's sort of appropriate that mobolisib is a JAK kinase inhibitor that was developed by a company that Andrew Wilkes founded in Australia, who was obviously the discoverer of the JAK kinases. And these inhibitors are used in the clinic for treatment of myelodysplastic cancer syndromes and also inflammatory conditions. And it's sort of amusing that one can watch the morning news on television and see ads for several of these JAK kinase inhibitors, including TYK2, which is a TYK2 inhibitor used for treatment of plaque psoriasis. So it's been a long road since the discovery of CML, which led to the first drug being approved, Gleevec, in 2001. But you can see there are many threads that were required: polyomavirus, RSV, and the Abelson murine leukemia virus, which all contributed to the discovery of tyrosine phosphorylation and its role in cancer. Now, I've never really worked on the JAK-STAT pathway. I've only had one paper in 1995, so now 30 years ago almost, on JAK signaling. But I thought I'd sort of, in the second half of my talk, discuss our work on pancreatic cancer, which involves cytokine signaling through the JAK-STAT pathway. Now, I'm sure you're all aware that pancreatic cancer is one of the deadliest cancers. It's almost always diagnosed too late, when the disease is already metastasized, and 5-year survival is only a miserable 13%. Now, why is it such a hard disease to treat? Well, it's a highly stromal desmoplastic tumor. There are nests of tumor cells surrounded by a dense matrix contributed by cancer-associated fibroblasts. This is a collagen matrix. It's poorly vascularized. And this is really a wound healing response against the tumor initiated by pancreas-resident stellate cells, which are activated into myofibroblasts. It's a highly immunosuppressed environment. Cytotoxic T cells are excluded, and immune cells such as tumor-associated macrophages, myeloid-derived suppressor cells are infiltrated into the tumor. And so it turns out it's a cold tumor, and immune checkpoint therapies have basically no effect in pancreatic cancer. And these protein factors secreted by the tumors, the cancer-associated fibroblasts and these myeloid cells and neutrophils that result in exclusion of T cells from the tumor nests. So about 12 years ago now, I became part of a Stand After Cancer Dream Team, and our goal was to try and identify soluble factors made by the stromal cells that might act on the tumor cells. And we already knew the tumor cells generated soluble factors like TGF-beta and PDGF that activate these quiescent stellate cells that are resident in the pancreas and lead to their activation and proliferation. And what we discovered was that the leukemia inhibitory factor cytokine is produced by the CAFs and acts on the tumor cells, as you'll see. So we approached this problem by growing CAFs, human CAFs in culture, and human pancreatic tumor cells like MIA-PACA2 in culture, and then harvesting serum-free conditioned medium and carrying out proteomic analysis. And this was largely the work of Ruojiu and Tian. At the time, a postdoc in Tony Paulson's lab, and Yu Shi, a postdoc in my lab. And we focused on growth factor cytokines and chemokines unique to the CAF fibroblast conditioned medium. And we found 3 members of our 6 family, IL-6, IL-11, and LIF. And we decided to focus on LIF because very little was known about its role in cancer. Its RNA levels are the highest in pancreatic cancer among the 20 human tumors surveyed by TCGA. And obviously LIF, like all IL-6 family cytokines, stimulates phosphorylation of STAT3 and tyrosine 705. But in one sense, even more importantly, LIF is a stem cell factor that elicits an invasive phenotype in the implanting embryo, and also induces an immunosuppressive microenvironment, which could be of importance for tumor progression. And in the published paper in Nature now, almost 5 years ago, we were able to show that the conditioned medium from the cultured CAFs can stimulate STAT3 phosphorylation. And if we knock down the LIF receptor, we see a significant decrease in the ability of the conditioned medium and LIF to induce STAT3 phosphorylation, and we could show that the signal induced by the conditioned medium is largely abolished by pre-treatment with a neutralizing LIF antibody. So this suggested that one of the factors made by the CAFs that activate STAT3 signaling is LIF. We went on to use the KPC genetic model of pancreatic cancer. We treated these mice with either a control normal IgG or a D25 anti-LIF antibody and stained for phospho-STAT3. And you can see strong staining in both the tumor cells and some of the stromal cells surrounding these tumor cells with phospho-STAT3 is a nuclear stain. And treatment with 25 mg per kg of the antibody 3 times a week strongly reduced the nuclear phospho-STAT3 signal. And we could show that a triple chemotherapy pre-treatment of these mice, that's nab-paclitaxel, a tubulin drug, gemcitabine, a nucleoside analog, and cisplatin, followed by treatment with gemcitabine and anti-LIF antibody, give a significant extension in survival of these mice. It's a pretty aggressive disease with an average survival time of around 50 days from birth. What we showed in other experiments was that not only does the neutralizing anti-LIF antibodies slow tumor progression, but it reduces the number of stem-like tumor cells and enhances the response to chemotherapy, suggesting LIF blockade could be of therapeutic benefit. Importantly, we could show that there is elevated levels of LIF in pancreatic tumor tissues from human patients. Many have very high levels of LIF. And also one could detect significant levels of LIF in the serum of pancreatic cancer patients. Note this is at a log scale here. So from these studies, we concluded that LIF can play a driver role in this mouse model of pancreatic cancer, that LIF made by the CAFs can act on the tumor cells, but probably also on other cell types, particularly the myeloid-derived suppressor cells. That LIF levels in human pancreatic cancer in serum and tumor samples correlate positively with progression. So this raises the question then whether LIF can be targeted therapeutically. Northern Biologics, a biotech company in Toronto with which I'm not associated, developed their own neutralizing anti-LIF antibody, MSC1, and carried out a phase 1 trial in highly refractory cancer patients, including 9 pancreatic cancer patients, and the trial was successfully completed with no MTD being reached, and 1 pancreatic cancer patient showed maybe stable disease. So the use of neutralizing anti-LIF antibodies obviously is one way to target LIF. One can also develop anti-LIF receptor monoclonal antibodies, and there are companies doing that. And one can also engineer the LIF receptor itself to generate a LIF trap protein. And I'll show you one slide here, because this work was done by our older son, Sean, when he was a graduate student with Jennifer Cochran at Stanford, and he took these 3 domains of the LIF receptor and carried out multiple cycles of random mutagenesis and yeast surface display to develop an engineered LIFR fused to an FC region that has about a 30-fold increase in affinity for human LIF, 30 picomolar affinity. And with our help was able to show then that this ELIF-RFC slows xenograft tumor growth of these human pancreatic cancer cells. So this is certainly another viable approach. There are efforts being made to develop small molecule inhibitors of the LIF-LIFR interaction. An EC359 has been reported to do this, and then I'll just show you one more approach that we have been using in collaboration with Ron Evans, and that is to block super enhancer-driven LIF expression using epigenetic inhibitors, in this case an HDAC inhibitor. This is the work of Gao-Yang Liang, a postdoc in Ron's lab, who showed that in the KPC mouse model that the combination of entinostat, a class 1 HDAC inhibitor, and gemcitabine, the nucleoside analog, gives a modest increase in survival of the KPC mice, but also reduces the tumor grade significantly. Now, it turns out the LIF gene scores as one of the top super enhancers in human pancreatic cancer tissue, as shown by Andy Lowe's group at UCSD. So these are genomic regions marked by high K27 acetyl, H3 K27 acetyl levels. And you can see these are the 10 top super enhancers. LIF is the top, and oncostatin M is the second highest super enhancer, and I'll get back to that right at the very end. So what Gao-Yang was able to show was that if you pre-treat human calves with entinostat, and then take the conditioned medium, that there is a significant decrease in the ability of the conditioned medium to induce phospho-STAT3. And you can see the anti-LIF antibody wipes out all these signals. And importantly, then entinostat treatment of the calves significantly reduces the level of LIF RNA and LIF protein secreted by these cells. So what happens in the treated mice? Well, it turns out that, as I told you, the tumors are highly immunosuppressed with exclusion of CD8 T cells. If you now treat the mice with entinostat, then you can begin to see infiltrating CD8 T cells. You can see that quantified over here. So we can conclude then that entinostat, HDAC inhibition, by targeting CAFs in combination with gemcitabine, which targets the tumor cells, can prolong survival in the KPC mice and delay tumor progression. And it appears to promote infiltration of CD8 T cells, raising the possibility that one could combine an HDAC inhibitor with a chemotherapy drug and possibly also with a checkpoint antibody. So what's happening with the neutralizing LIF antibody from Northern Biologics? Well, it was acquired by AstraZeneca, renamed AZD0171, and they are carrying out it's an ongoing phase 2 trial which they've combined AZD0171 with durvalumab, their anti-PD-L1 and checkpoint antibody gemcitabine and nab-paclitaxel in advanced pancreatic cancer, and we've not heard any results yet, but this is supposed to be unblinded sometime in 2024. So I think we're all keeping our fingers crossed that this might actually be a viable approach. So to end then, I can tell you what we're currently doing. As I already told you, several other LIF-related cytokines have been implicated in cancer, such as IL-6. And oncostatin M, which was very high as a super enhancer driven gene, is an IL-6 family member that utilizes both the LIF receptor and a dedicated OSM receptor. And we've begun to investigate oncostatin M's role in pancreatic cancer. I should point out that its RNA levels, oncostatin M RNA levels, like those of LIF, are quite elevated in pancreatic cancer compared to the normal pancreas. And it has been implicated in several types of cancer, including glioblastoma as well as pancreatic cancer. And the ligand in this case is highly expressed by the tumor-associated macrophages and not the CAF so much. And it signals through both the LIF receptor GP130 and oncostatin M GP130 to drive JAK-STAT signaling. And so that's sort of summarized here. Oncostatin M has its own receptor as well as using the LIF receptor. And the oncostatin M receptor also is liganded by IL-31. So ultimately, all of these drive JAK-STAT signaling, which is, of course, the whole theme of this meeting. And I'm excited finally to be able to do a little bit of work on JAK-STAT signaling. So I'll end here by thanking the people who did the pancreatic cancer work, spearheaded by Yu Shi and Ruzhen Tian, the help of several collaborators from UCSD and other institutions for the clinical samples, my son Sean, and other collaborators at the Salk Institute. And with that, I'll Thank you all for listening, and I'm happy to take any questions if there are any.
就是现在呢，我们要成立这样的一个研发联盟，这个也是为下次会议做准备的。